Can you see my screen, everyone?
Yes.
Okay, I'm going to start from where we stopped in class. Last time we were in class, the last thing we discussed was the adaptations of the alveoli. I wanted to show you this picture. I think I showed you this in class. And the movement of the oxygen from the alveoli to the surrounding capillaries and the carbon dioxide in the opposite direction. And we said that alveoli, they have adaptations. Their function is gas exchange, and they're very good at gas exchange. And the reason they are very good is that they are very thin, so this helps in diffusion. It shortens the diffusion distance. They're very close to a good blood supply, and this maintains a high concentration gradient for faster diffusion. This sentence, I focused on this sentence because we said we want to understand it. Maintain a high concentration gradient means I have so much oxygen in the alveoli, little oxygen in the capillaries. That is why the oxygen will always diffuse from high concentration in the alveoli to low concentration in the capillaries. And this high supply of oxygen is provided due to good ventilation. Ventilation means good aeration. And also I have high concentration of carbon dioxide in the capillaries, and this high concentration of carbon dioxide is due to good blood supply. The blood is supplying the carbon dioxide. So we have high concentration of carbon dioxide in the capillaries and low concentration of carbon dioxide in the alveoli. That is why the carbon dioxide always moves from this direction, from high concentration in the capillaries to low concentration in the alveoli, and then the carbon dioxide is exhaled. Okay, so these adaptations are supplements and they are important. Alveoli have a large surface area. They are so many in number. We have millions of alveoli, and this increases the surface area. Of course, the more the surface area, the faster the fusion. Number 4, they have a good supply of oxygen. They are well ventilated. We already said that, which maintains a concentration gradient. They have moist walls, alveoli, they're moist, there's moisture, and this moisture helps the gases to dissolve, and of course helps in diffusion. All this helps in diffusion. And also the alveoli are permeable. Like the capillaries, they have pores. Alveoli, if you draw them, you can draw them like this with pores, and these pores, of course, help in the diffusion of gases. Okay, so this was the last thing that we mentioned. Fine. All these features are found in gas exchange surfaces. Gas exchange surfaces include alveoli in mammals, and in fish, they are the gills. Fish, they don't have alveoli, they have gills. Fine. Now, the last part that I want to explain in this lesson is the practical part, which is very important. Now, counting the number of breaths per minute, or the breathing rate. The breathing rate is the number of breaths per minute, just like the heart rate. This is per minute. If you remember the heart rate, we count the number of heartbeats per minute, and we took that we do this either using a manual method, using our fingers. We will put our fingers on an artery because arteries have a pulse, and we will count the number of pulses per minute. Or if we are in a clinic, we can use a stethoscope or a digital pulse using electronic devices or the electrocardiogram. If you remember, that will give us an image on a screen. And every wave is a heart contraction. This is the heart rate. Why am I reminding you of the heart rate? Because it is very similar to the breathing rate. The breathing rate is the same thing. I want to know how many times you will breathe per minute. And I use this, of course, in investigations. There are two methods. There is an accurate method and an inaccurate method. The accurate method is what you see here in the picture. This is the device. It is called the spirometer. The spirometer is an electronic device, as you can see. Part of the device will be in the mouth of the individual, and the other part is digital or connected to a screen. And it will give you the number of breaths per minute. Of course, this is accurate. What if you don't have this spirometer? What will you do? You will count the number the chest will move up and down in 1 minute. Of course, this is not accurate. But this is what you will do if you don't have a spirometer. You don't have this device. You will have to put the hand or your hand on the chest of the individual and count the number of times the chest will move up and down in 1 minute. This is not accurate, but this is what you will do if you don't have a spirometer. And again, If you will do any investigation that involves exercise, you will measure the breathing rate before exercise, which is the starting breathing rate. And then you will measure the breathing rate after exercise, which is the final breathing rate. And what do you calculate to know the change in breathing rate?
Percentage change.
Exactly. The final breathing rate minus the starting breathing rate over the starting breathing rate times 100. Very easy, and it is the same thing as the heart rate. And of course, we mentioned that exercise will increase the breathing rate because we want more oxygen for aerobic respiration. We want more blood flow. We want to remove the excess carbon dioxide. We said all this many, many times. Now we have a table here that is very important. This is also supplement, in which they ask you for the percentage of the gases moving in and moving out and the reasons for this difference. So this table is very important. Oxygen, how much oxygen moves in? From 20 to 21%. And the oxygen that is exhaled, of course, less oxygen goes out, 16%. Now, why? Where does this oxygen go? I don't even need to read this part. The oxygen is used for what? You tell me. It's so easy.
Aerobic respiration.
Very good. Carbon dioxide. Carbon dioxide you take in when you inhale 0.04% carbon dioxide, and what comes out or you exhale more carbon dioxide, 4%. And why? Why do you exhale more carbon dioxide? Tell me.
Yes. Waste product of aerobic respiration.
Very good. Okay. So this column is very easy, but these numbers, you have to memorize them. You will see questions asking you specifically about these numbers. Okay. Now, nitrogen. Nitrogen is a gas that is found in the air. We don't use it. It is completely unreactive. And actually, most of the air is nitrogen gas. 78% goes in, and the same percentage goes out because it is not used up. It is completely unreactive. Now, the amount of water content. The amount of water content, it's not a number, but the water vapor that enters our body is always less than the amount. Excuse me, please put down the voice. The amount of water vapor that is inhaled is always less than the amount of water vapor that is exhaled. Now, why is that? Always when we exhale more water vapor for several reasons. Because the gas exchange surfaces, they are moist, we said. There is lots of moisture in our respiratory system. And this moisture evaporates as we breathe out, and it comes out with the exhaled air, okay? And also, another reason that you can add to this box is that the water vapor is actually what? Isn't it a waste product? Isn't it a product of respiration? Respiration is food plus oxygen to give us what? Carbon dioxide and water. This water comes out of the body in so many different ways. It comes out in the form of sweat and also as water vapor. You exhale it, okay? So the water content is easy because there are no specific numbers, no numbers for the water or the temperature. The numbers are only for two things, the oxygen and the carbon dioxide. But the water vapor that always moves in is less than the amount of water vapor that goes out. Now, why do we have it variable here in the biology books? Because it depends on the surrounding. Sometimes you will have more water vapor on one day than another day. But it is always less than the amount of water vapor exhaled for two reasons. Because water vapor is a waste product of respiration, and because you have lots of moisture in your respiratory system, and this moisture evaporates as you exhale. Now, the temperature, think about it. You don't even need to read it. The temperature of the air going in, is it colder or warmer than the air going out? It's usually what?
Colder.
The exhaled, yes, it's colder. Usually the air that comes out of your mouth, it's warmer. It takes from the temperature of the body, okay? So whatever air that comes out of your mouth, it is always higher or always warmer. And why do we have it variable here? Because again, the temperature of the air going in depends on the surrounding temperature. So this table is very important and it is very easy. The only numbers that you need to memorize are the oxygen and the carbon dioxide. Of course, you have more oxygen in and less oxygen going out because the oxygen is used in aerobic respiration. The numbers, 20 to 21% oxygen goes in and 16% oxygen goes out. Carbon dioxide, of course, you have less carbon dioxide inhaled and more carbon dioxide exhaled. Why? Because it's a waste product. You need to memorize numbers. 0.04% carbon dioxide in and 4% carbon dioxide out. Nitrogen, the percentage does not change because it is unreactive. We don't use it. Water content, always the air coming out, the air exhaled is always saturated with water. Okay, because it's a waste product of respiration and because lots of water evaporates from the moist walls of our respiratory system. And also, the air coming out of our mouth is always warmer because it takes from the temperature of our body and it is warmed as it moves through the respiratory system. Any questions over this table? They ask you over this table in paper 4 and paper 6, so it's important. Any questions? Now, we will continue paper 6 material. Testing for carbon dioxide, I think most of you know, it's very easy. We have two tests. We have the lime water and we have the hydrogen carbonate indicator. The lime water is very easy. I'm sure you took it in chemistry. Lime water is transparent. When you bubble in carbon dioxide, what is the color change?
Milky.
Very good. It will change from transparent to milky, from colorless to milky. Good. Now we don't have even to know the reason why, but I think I mentioned it before because the carbon dioxide dissolves in water and forms an acid known as carbonic acid. And this acid will change the color of the lime water from colorless to milky. Now, the hydrogen carbonate indicator, I think you saw for yourself that they ask a lot of questions about the hydrogen carbonate indicator. Sometimes they give you the color, sometimes they don't. So you need to know the color. Now, this indicator helps to indicate the presence of carbon dioxide in the surrounding liquid. If we have normal levels of carbon dioxide, atmospheric levels means normal or average levels of carbon dioxide, the color is orange. Remember the color. High carbon dioxide, the color is yellow. Low carbon dioxide, the color is purple. Okay? Now, this is usually very common in plants. I think we saw it. In which process, photosynthesis or respiration, does it cause an increase in the carbon dioxide levels in the surrounding water? In other words, if I have water and I have plant inside water, it is an aquatic plant, and I have in this water hydrogen carbonate indicator. When will it change to yellow? Because of the high CO2. When there is more photosynthesis, respiration, yes, okay. Some students, they get confused because they tell me that the carbon dioxide will increase inside the plant cell because the plant cell will absorb CO2 to do photosynthesis. Okay, but we are not looking at the carbon dioxide levels inside the leaf. We are looking at the carbon dioxide levels in the surrounding water, When the CO2 levels are high in the water, this is because the leaf will respire and will release carbon dioxide. When the carbon dioxide levels are high in the water, it will change yellow. So don't get confused. This question, I think you saw it a lot. Okay? Now there's another test for water vapor that is not very common, but we need to be on the safe side. And I also think you took it in chemistry. There is also a paper that is very similar to the litmus paper. It is called the anhydrous cobalt chloride paper. You don't need to memorize the name. I think they will always give you the name and the color change. This paper is blue in color, and when it is exposed to water vapor, it will change to pink. It's not common, but it is in the Cambridge book, so we have to mention it. What's important is the hydrogen carbonate indicator. Don't forget it. The lime water, you all know it. In the presence of carbon dioxide, it will become milky. The hydrogen carbonate indicator, the normal carbon dioxide levels, makes it orange. High CO2, yellow. Low CO2, purple. Please memorize the colors. Okay, another practical investigation. You have here 2 test tubes, and these test tubes are set up in this manner. You have one long tube coming from outside, that is inserted into lime water. This is lime water. And you have another setup in which the longer test tube is coming from a mouthpiece, and you will breathe out. When you breathe out, what is the gas that will come out as you exhale?
Carbon dioxide.
Carbon dioxide.
Carbon dioxide. Okay, the question will ask you that you will breathe out at the mouthpiece. What do you expect will happen in the color change at test tube A and test tube B? Which test tube will change from colorless to milky?
B.
Yes, B. Let's look at B. It's very simple because you will breathe out here. Let me remove all that. You will breathe out here and the air will move down, which contains CO2, okay? This is exhaled air. It will move down into the long test tube and into the lime water. So the lime water here will be exposed to CO2 and the color will change from colorless to milky. So tube B, this is the mark scheme. Tube B will become cloudy or milky. Now tube A, the longer test tube is connected to the outside. The outside contains less carbon dioxide. So it will just stay clear. There will be no change here in test tube A. That's it. Okay? They can ask you what are the safety precautions. Of course, you will put your mouth here. Is this a safe experiment?
No.
It's not. Why? Why would you avoid doing such an experiment?
It can be contaminated.
Of course, this mouthpiece can be not clean. You can break it if you like. You can break it. Of course, this is not very common, but you can. Maybe this mouthpiece, someone else put their mouth there and you don't want to. Or you can suck in the lime water. So these are the safety precautions that you need to change the mouthpiece. Don't suck too hard and don't break the glass. Very easy experiment. Also, this is not common. This is common. This is important. And it clearly says in your syllabus that they want this experiment, the effect of temperature on the respiration of yeast. Now, before I start, yeast, I would remind you again for the 10th time, yeast is unicellular fungi that can respire both aerobically and anaerobically. A lot of you assume that yeast respires anaerobically. No, it can respire both, depending on the availability of oxygen. Now we want here to investigate the effect of temperature on the respiration of yeast. So what is the independent variable? What will I change?
Temperature.
Temperature. And as you can see in the picture, what do I do to change the temperature? What do I use?
Water bath.
Water bath.
Very good. Water bath. The picture is very clear. You have this picture in your notes. You have two test tubes and I'm going to put equal volumes of yeast in one test tube. And in the other test tube, I will put a solution of glucose. I will put it in the water bath for 5 minutes until it reaches equilibrium or it takes the temperature of the surrounding water. And then I will mix them together and I will put the test tube back into the water bath. And I'm going to add a dye, a methylene blue or DC-PIP dye. You don't need to memorize the dye, but they will give you this piece of information in the question. They will add this dye, whether methylene blue or DC-PIP, which is blue in color, and this dye will change from blue to colorless due to the release of carbon dioxide. So how will you measure the rate of respiration? Why is there a stopwatch here? What do you think?
To see the color change ولا يعني
Yes,
to
you will
estimate the
see, you-
solution to change color.
yes, you will see a color change because carbon dioxide will be produced, but you will see the color change. Okay, what if the color changes from blue to colorless very quickly? Will this tell you that the rate of respiration is high or low?
High.
High. And how can you measure the rate of respiration?
Over time.
Over time. In this case, there is nothing that you can divide over time. How can you modify the experiment in order to divide a factor over time and calculate the rate of respiration? I can change the whole experiment so that I can get, let's say I'll help you. The volume of gas produced over time. I will not here look at color change. What is the gas being produced?
نحط الفوليوم of gas اللي هو CO2 على time.
Very good. Okay, so let me repeat it because maybe some of you are confused. We are doing an experiment. We want to measure the rate of respiration. So the rate of respiration is my dependent variable or my results. And what am I changing? I'm going to change the temperature. So I'm going to do this experiment and I can modify it. I'm going to get a water bath and I will get different thermostatically controlled water bath, one that is cold, one that is warm, one that is hot. Please give examples. And I'm going to put 2 test tubes, one containing my yeast and one containing my glucose, equal volumes of yeast and glucose and equal concentrations each time. And I'm going to keep it in my water bath for 10 minutes so that it takes the temperature of the surrounding water or it reaches equilibrium. After 10 minutes, I'm going to mix the 2 test tubes together. And I can do 2 things. So here I have the mixture. I have the yeast plus the glucose. Now the yeast, the moment it will come in contact with the glucose, it will take this glucose and break it down. This will be aerobic respiration because I have a supply of oxygen. I have a gas that will be produced. What is this gas?
Carbon dioxide.
Carbon dioxide. If I want to modify my experiment and know the rate of respiration, I can connect this test tube into a graduated syringe and measure the volume of the gas produced over a fixed amount of time. And in that case, I'm going to calculate the what. When I divide the volume of the CO2 over the time, this will be what? What am I measuring? The rate of respiration, okay? But maybe they don't want the rate of respiration. They will do a simpler experiment. This is a simpler one. I'm not going to measure the volume over time. I'm going to put a dye, that's it. And I'm going to measure the time for the dye to change color. And the shorter the time, I will assume that the respiration rate is fast. So they can give you this experiment and ask you to modify. I gave you all options. Do you understand? The experiment that is provided here, they did not measure the volume of the gas. But if they ask you to modify, you will tell them, instead of putting a dye and just looking at the color change, I'm going to connect my test tube to a graduated syringe that will measure the volume of the gas produced over a fixed amount of time, and this will help me calculate the rate. Okay, so do you understand what was done here? Any questions? I will put the yeast in one test tube and the glucose in another test tube. I will put it in a thermostatically controlled water bath. I will wait for 10 minutes until I reach equilibrium. What does reaching equilibrium mean?
The temperature
Both
inside is
systems
equal to
have
outside.
the same
Yes,
temperature.
they take the temperature of the surrounding water. I have another question for you. Why did I start by separating the yeast and the glucose? Why don't I just put them in
Because
one test tube from the beginning?
They will react together.
Exactly. I don't want them to react just yet. I want to first reach my desired temperature and then mix them so that after I reach my desired temperature, I will react. Either I'm going to add a dye and measure the time needed for the color change from blue to colorless, and don't worry, this dye does not affect the yeast cells, or I can connect it to a graduated cylinder, which is going to be a modification so that I measure the volume of the CO2 produced over time. Okay. Now, another experiment, which is very close to it, investigating the effect of temperature on anaerobic respiration in yeast. Again, we will do something very similar. We will get the yeast and we will put it in a test tube. And we will get the glucose and we will put it in another test tube. And we will put it in a thermostatically controlled water bath. Cold, warm, hot. We will wait for 10 minutes until they reach equilibrium. And then I'm going to mix the yeast and the glucose together and put them back in the water bath, but I'm going to add a layer of oil. Why did I add a layer of oil?
to prevent
To
oxygen.
prevent the oxygen
Very
and
good.
the CO2 from escaping.
Excellent. I want to prevent the entrance of the oxygen because I want this yeast to respire anaerobically, okay? And any CO2 also will be prevented from escaping. So when I add a layer of oil, I'm going to add a layer of oil to prevent the oxygen because I want the yeast to respire anaerobically. Okay, now what happens? The yeast will break down the glucose. Glucose will be broken down anaerobically to give me what? Alcohol and CO2. The yeast here is above the arrow. This CO2 will be delivered using a capillary tube, and it will enter another test tube containing lime water. What will happen to the color of the lime water?
Turn milky.
Very good. And we will count the number of bubbles. Is this accurate?
No.
No.
Of course, it's not accurate because the bubbles we can miscount. They can be of different sizes. So how do I improve?
Graduated gas syringe.
Very good. I think now you became experts. Graduated syringe over time. So when you see an experiment and they have a layer of vegetable oil, then know that they put this layer of oil so that the respiration is anaerobic and the CO2 does not escape. When you see an experiment without a layer of oil, then the respiration here is aerobic or anaerobic?
Aerobic.
Aerobic. Very easy experiments. They come in paper 6. And then they will give you a graph and they will tell you that this graph is showing you how the temperature change will affect the rate of respiration and describe and explain. Okay? Again, when you are asked to describe and explain any graph, divide the graph into 2 parts or 3 parts, depending on the shape of the graph. Part A, part B, part C. Now help me. How will you describe A? Describe and explain. Now we did this.
Increasing the rate of the respiration as temperature increase.
Okay, this is the describe part, and you will give numbers. Who wants to answer? Someone raise their hand. Don't forget the numbers, okay? Now they explain. Explain A. I want someone else to answer. Why, when the temperature increases, the rate of the respiration will increase? Why? What do we say here? Don't forget, respiration is catalyzed by enzymes. Enzymes are in the yeast. Exactly what we took in the lesson, enzymes.
They have more kinetic energy, they move faster, more collisions.
Perfect. More collisions. What else? Let's write everything. More what? More
Substrate complex.
Excellent. Enzyme substrate complex. More product is produced. Very good. Now, what do we say at B?
The peak of the reaction.
Very good. The
Optimum
optimum temperature, peak of reaction, fastest. Everything is faster here. What is happening at C?
The nature of the enzyme.
Yes, and we will elaborate. We will say the active site will not be complementary anymore to the substrate. It will not fit together. They will not form enzyme substrate complex. No successful collisions. I think you memorized this by now. We're done with the lesson. That's it. This is what I wanted to explain today. Here, I just want to show you some pictures. This is the dough rising and the fermentation of yeast when we use it in bread making. This is the trachea. And this is the esophagus. These are the C-shaped rings because you did not see those pictures in class. What else? The anatomy, I think you're very familiar with it now. Don't forget the larynx above the trachea, the voice box. And I think this is clear. This is going to move, CO2 going out, oxygen going in. We mentioned this, yes, because this is important. The advantages of exercise on heart rate. It's related also to what we're studying. Exercise will prevent you from having blocked arteries, prevent the formation of a blood clot or a thrombus, lowers blood pressure, lowers cholesterol, lowers fat, reduces atheroma. Atheroma is a blood clot, and it helps you lose weight and also increase cardiac output. The heart muscle will become stronger. It will lower the resting pulse. By the way, this is copy-pasted from a mark scheme from one of the questions. I did not write it. Okay, I think we answered those questions, didn't we, or we didn't? I think we answered these questions. Did we answer these questions? In the first online? Can someone give me an answer? Because if we
No.
didn't solve them now, we did not?
I don't think so, لا.
Okay, let's leave those for now. We will solve them. I want to solve this one. I just gave you the answer. The diagram shows an experiment to investigate the respiration of yeast. Oil prevents oxygen entering the glucose and yeast suspension. So this is the thin layer of oil. If no oxygen is present in the glucose and yeast suspension, what will occur? A, ethanol will be produced and the lime water will stay clear. Is A right or wrong?
Wrong.
Wrong. B, ethanol will be produced and the lime water will go cloudy. What do you think? B is right or wrong? In anaerobic respiration, what is the result? Glucose will not react with anything. It's
صح.
anaerobic. Yes, is correct. Okay.
True.
And C, is lactic acid going to be reproduced?
No.
No, lactic acid will not be produced. Okay, let's look at this question. What is the sequence of structures through which a molecule of oxygen passes from the air to the blood of a person? So here you have to know the anatomy and what is the first structure that the oxygen will pass through.
Larynx.
Yes, the first thing that comes in contact with the air is the larynx. So you have the larynx. So the answer is C or D. It is not A, it is not B. Tell me, is it C or D? Is it the larynx and then the bronchiole, or the larynx and then capillary?
D.
No, the capillary
C.
is the last structure.
Bronchial.
Yeah, with the
Bronchial.
bronchiole. Okay, we have
C.
bronchi and then the smaller bronchiole, and then alveoli and then capillary. Okay, now let's look at this question. This question is showing you here small insects. Any insect, any small animal in an experiment, then this animal is doing what?
Respiration.
Exactly. Always put this in mind, respiration. If it was a plant and there is sun, then it will be doing photosynthesis. And actually, the question is telling you, a student is investigating aerobic respiration. Soda lime absorbs the carbon dioxide. So soda lime here will absorb the carbon dioxide that is being produced. If the wood lice are respiring aerobically, aerobically means they are also using up oxygen. So the wood lice will take the oxygen and will give out carbon dioxide, and the carbon dioxide will be absorbed by the soda lime. They will ask you, what will happen to this drop of liquid? In which direction it will move? Towards the wood lice or away from the wood lice? What do you think?
Towards.
Yes. Why towards?
Because if there is any type of air, it will push the drop to يعني against the wood lice. بس ما فيش أي air فit will be يعني pulled.
Yes, you are correct. Okay, this question can also appear as a paper 4 question. Now, how will you answer it if it's a paper 4 question? You will say that the wood lice will respire anaerobically, releasing what? Using oxygen. So the oxygen will be used up. The oxygen in this small space will be used up. And it will release carbon dioxide, but the carbon dioxide will be absorbed by the what? By the soda lime. So inside this small space, like Selma said, there will be no air. The best way to say it is that there will be negative pressure, because all the gases are being absorbed. And this is going to pull the drop of liquid towards the wood lice. So let us see. If the wood lice are respiring aerobically, what will happen to the drop of liquid? A, it will move towards the wood lice as oxygen is used up and carbon dioxide is released, or it will move away. No, I will not choose B. It will not move. No, C is wrong. It will not move as oxygen is used up and carbon dioxide is released. What is the answer? What do you think? It will not move, or it will move towards the wood lice? Give me an answer.
A.
Yes. Okay. Now, describe similarities and differences between anaerobic in yeast and aerobic respiration in humans. This is a nice essay question. Aerobic respiration in humans. The best way to answer this question is to write the formulas that you already know and look at the similarities and the differences. Let's look at the anaerobic respiration in yeast. Who will remind me what's the equation? Remind me. I forgot. Wake up with me.
Glucose is breaking down by enzymes
Very good. Yes.
to give ethanol and carbon
Yes.
dioxide and energy.
Okay, good. And we are comparing that with aerobic respiration in humans. Can you remind me what is aerobic respiration in humans?
Oxygen
Glucose,
plus water
oxygen.
equal
Plus water. Stop. Oxygen plus
Glucose,
water.
oxygen,
Yes,
glucose.
because someone said oxygen plus water. Okay, will give me what? Carbon dioxide and water and energy. Okay, now what are the similarities? Huh? Yes, my dear.
Anaerobic respiration.
Yes, anaerobic respiration in yeast. It's okay. Okay, so tell me, what similarities do you see and what differences? At least give me two.
CO2 and energy are a product in both.
Yes, energy is produced in both. But what are the differences? Which one do you think will produce more energy?
The aerobic.
Yes. Okay. What else? Give me another similarity. What is broken down in both cases?
Glucose.
Glucose.
And also, which gas is produced in both cases?
Carbon dioxide.
CO2.
Yeah. Okay. So if you write the equation, you will know how to figure out an answer. Now, what are the differences?
There is no oxygen in anaerobic
Yes, there is no
yeast.
oxygen here. Another difference.
No water is produced.
No water is produced in the anaerobic ethanol is produced. It's easy only when you have the equations in front of you. Both require anaerobic. Okay, easy question. Fine. I'm done with the respiration. Okay, I'm going to solve with you the last 10 pages of paper 6 plants because starting tomorrow, I'm not going to solve any more paper 6 plants. Enough. We solved a lot of paper 6 plants because a lot of you are complaining that it's too long. So I think I solved 20 pages with you in class and some classes, and I will solve the last 10. And then the amount of the number of pages in the middle, you have to solve them as homework. You don't want to, it's your problem. Okay, now paper 6 plans is the best exercise for paper 6. Any question about paper 6 will be asked in paper 6 plans, investigation, graphs, etc. So open paper 6, please, at page 244. Let's solve together. The last 10 pages will take exactly 15 minutes, and then I'll let you go. The first question here tells you that figure 2.1 shows the lower surface of a leaf from a dicot plant on a 1 centimeter squared grid. Make a large labeled drawing of the leaf. I'm going to draw it with you. And please, if you have the paper 6 in front of you, draw it, okay? Now, how do I draw that? I'm going to get my pen. I want an exact copy of this leaf. And even you have to label it. I'm going to use more than half the space available, no broken lines, no shading, and I'm going to make it exactly similar. And I have to provide details. You know what I will do before I draw? I can count the number of veins. I can see 1 here, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so I'm going to do something like this. And the middle vein is thick. So I'm going to draw a middle vein. Okay, please put in mind that it's not easy to draw on my laptop. And then I'm going to draw the leaf. No, this is not nice. See? This is not nice. I'm going to erase that. And then I'm going to draw. Okay, it's not very pretty, but I'm trying my best to do it on my laptop. And I have here nearly 10 veins. 3, 4, 5, 6, 7, 8, 9. Okay, now of course it's not lovely, but I want to show you that you have to use more than half the space available, no broken lines, no shading at all, and you have to exactly replicate what you see. Now I want someone to give me at least 2 labels. What will you label this?
The midrib and the veins.
Yes, it's the midrib. Okay, midrib, good girl. And you can make an arrow here or write vein. And if you also label the midrib as the vein, they will consider it correct, okay? This is the petiole,
Tip.
the part that is connected to the stem. This is the edge or the edge here. Here, the leaf is called the lamina. Now, I want to show you the answer. This is page what we said. 244. Why do I want to show you the answer? Because you have more than one picture of the, what's this called? Of the leaf in your notes. You have actually that picture in your notes. So please, and next to it, it says clearly know how to draw the leaf, because drawing the leaf was asked in paper 6 more than once. It was asked in this question, and it was asked in this question, in more than one question. So drawing the leaf also was important. Please, okay? Image from the internet, we're drawing this. So more than once they ask you to draw the leaf. So practice it, it's very easy. Good? Now here, there is a drawing here that I got from one of the students, but I will object over this drawing. It's very good. I'm going to object on this drawing. This drawing will not get a 5 out of a 5. Can you tell me why? This one. Number one, because the leaves are not smooth.
Sharp.
This leaf, okay, it's not
It's
smooth.
very wide.
It's very, no, it's wide. This is good. The midrib should be represented by 2 lines. So I'm going to change this next year, and I'm going to choose one of your pictures if they are very good, and I'm going to put it in an answer. Okay? Now let's go back to the question. So it should look like something like this. Calculate the surface area of the leaf. Okay, how do I calculate the surface area of the leaf? And again, this question appeared more than once. Calculating the
Count.
surface area of the leaf using a grid. What do you do?
Count the boxes.
Very good. I'm going to count the boxes, and I'm going to show the examiner that I'm going to count the boxes. How do I show the examiner? I make a check mark. And the easiest way to do is this: 1, 2, 3, 4, 5. This is occupying more than half the box, so I will include it. 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28. This is half, and this is half. So half and half, this will be 29. This is 30, 31, 32, 33. This is half. And let's say this is half. This is 34. And this is 35, for example. You can put the numbers directly on the box, which is going to make it very easy, or you can do a check mark, whatever, but you need to show the examiner that you counted the number of boxes. So in my case, I will count and I will find that they are around 34, and don't forget the unit. What's the unit?
Where unit?
Centimeter square.
Centimeter squared, because this is the surface area. Okay, so it's from 32 to 34. Very good. Describe the method that you used to determine the surface area. What did you do? You will describe what you did. So the answer here is from 32 to 34. Write it down. What did you do? Describe. Very simple.
Count the squares.
Count the squares, but I see here it's out of 2 numbers, so elaborate a little bit more. When I see that the leaf occupied less than half the square or more than half the square, I'm going to count it as 1. Okay, so I'm going to count. Write it down with me. The number of squares, and if the leaf occupies more than half the square, it will be counted as 1. Okay, now again, counting. Can you please mute, please? Excuse me. I'm going to mute you. I'm sorry. Okay, things are okay now. Now, this question also is repeated, which is counting or finding the surface area for a leaf or any irregular object. We use a grid, okay? I want to show you that it also showed up in one of the questions before, in more than one question. In which page? Let me look for it. And I actually added or showed you the grid in the answer. Either they will be nice and they will give you the grid, and they will tell you to count the number of boxes and find the area, or they will ask you, how can we find the surface area of the leaf? See, in this question, on page 229, which exam? November 2019 exam. So on page 229, I want to show you a different question. A student suggested that measuring leaf area is better than measuring the width of the leaf. Describe how the area of a leaf could be measured. So here, I'm recording. You don't need to record anything. So here you have a question that is not giving you the leaf and not giving you a grid. They expect you to know how to calculate the surface area of the leaf. Fine. So this is the answer given. You will draw or trace the leaf on a grid and you will count the number of boxes. And I provided those pictures for you so that you can imagine the answer. This was not in the mark scheme. I gave you those pictures so that you can see what they mean, so you understand what they mean. Okay, this is the grid. So expect either they will give you the grid and they will ask you to count, or they will ask you, how do we calculate the surface area of a leaf or any irregular object? You will tell them, I will get a grid and I'm going to put my leaf on the grid and draw an outline and then remove my leaf and then count the number of boxes. Or I can get my leaf and put a transparent grid on top. It's the same thing. So this is a new question, mark it so that you revise it before paper 6. Okay? Now let's go back to where were we? We were on which page? Yes, we were on this page, page 245. Figure 2.2 shows the upper and the lower surfaces of the same leaf of another plant. Now describe two ways visible in figure 2.2 in which the upper surface of the leaf is different from the lower surface. This is the upper surface and this is the lower surface. And don't tell me I don't find the difference. I want two ways in which the upper surface is different. Quickly. Very easy questions. We should not be wasting any
The
time on
midrib
those.
is
veins.
less transparent.
Okay, you can say exactly what you said can be written, or I'll write it in a different way. The veins are less prominent, are less obvious, for example, or less clear, or less, I can see them less. Another difference, the upper surface, look
Darker.
at the color. Yes, it's
Smoother.
darker. It's darker. Smoother, you have to feel it. The differences, they have to be visible. Okay, so the upper surface is darker and the lower surface is lighter. Okay, I'm going to move quickly so we finish. Now, a student investigated the effect of different wind speeds on the rate of transpiration. I will stop here. What's the independent variable?
Wind speed.
Yes, wind speed. And what are my results? This is my independent variable. What are my results?
Rate of transpiration.
Yes, I want everyone to participate. Make sure that you understand, even if you're not speaking out. The student took 5 leaves from the tree and weighed each of them on a balance, and then each of the leaves was then hung from a different piece of wire. Fans were used to blow air at different speeds over each leaf. After 12 hours, the student weighed each leaf again, and the results are shown. So this is the mass at the start, and this is the mass at the end. Of course, they will tell us calculate the loss in mass. How do I calculate the loss in mass? Quickly.
Minus.
Exactly, I will subtract. They did not say percentage loss. They said only loss. So for example, this one, I'll get 3.8 minus 5.7. Now, what's important is that all the answers should be to 1 decimal place, because I have to follow the same format of the numbers in the table. All the numbers should be to 1 decimal place. Okay, quickly do it, quickly, because you will use that to plot your graph. I'll give you the answers so we don't waste time. We said you need a calculator, page 246. Here, okay. So this has been done for you, and you have all the answers. And see, this is from the mark scheme, all values to 1 decimal place, okay? Now they will ask you to do what? To plot a graph. Now this is very important. Some of you are very good at graphs and some of you are still not good at graphs. And this is not an option because graphs are at least out of 4 to 5 marks. So we don't want to lose such a big number. By the way, losing 4 marks can affect 1 whole letter grade. Like if you lost 5 marks or 4 marks on a graph, instead of taking an 8, you will take a 7. It is that serious. So please consider that. Now on this graph, what do I plot on the x-axis? Tell me. What is my independent variable we said?
Wind
Because
speed.
when
Wind speed.
Different wind speeds. Okay, there are no units. No, there are units here. The wind speed, meter per
Meter per
second.
second.
So of course, I'm going to label it clearly. So I'll write here wind speed. Do it with me. Wind speed, meters per second. Good. And then what do I put on my y-axis?
Rate of transpiration.
Okay, what did I calculate? Because we
Loss in mass.
loss in mass. Yes. Okay, loss in mass. Okay, so I'll put here loss in mass in grams. Loss in mass. Do it with me. Loss in mass between brackets. I'll put G between brackets. Okay, I said it so many times. We will first put a 0. Make sure first, as long as all our values are positive, so the 0 will be here. Okay, and then I count. This is 1, this is 2, this is 3, this is 4, this is 5. I make a mark. And this mark is very important. Don't put a number without a mark. Again, I'll do it with you. 1, 2, 3, 4, 5. Okay, and I'll do the same thing on the y-axis before I start anything. This is very important. 5 boxes mark. Okay, now we'll count how many boxes do I have on the x-axis. I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I'll go to my table. I'll go to the wind speed. The maximum value is 8. I want 8 to be distributed over the x-axis so that it occupies more than half the graph. So I want the 8 to be here, or 10. Okay, how will I distribute that? Let me do it. 2, 4, 6, 10. Okay. I do here 2, 1, 2, 3, 4, 6. Okay. I think this is the best way. Every 3 boxes is 8. What do you think? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12 divided by 8. What's 12 divided by 8? Anyone has any
1.5.
other options? 1.5. No, I will take 3 boxes, and every 3 boxes will be, or I'll give them a value of 2, so that the 8 will be at the end of the x-axis. Good? Anyone has any other ideas? If I put the 2 here, some of you will give me this answer. If I put every 2 boxes 2, like every 10 small boxes 2, 2, 4, 6, and 8. You might lose a mark for that because all this was not used. Okay, so this is something we want to avoid. So we
Miss,
want the 8. Yes, my dear.
the question after this one asks for the mass loss at 10 meters per second. So there has to be values after 8.
Let me see. Use your graph to determine the loss in mass at a wind speed of 10 meters per second. Show on your graph. This is very good. Now, good that you spotted that. If this question was not there, you will have to have the 8 at the end of the axis. But she noticed something. She said that the next question here, they will ask you to use your graph to determine the loss in mass at a wind speed of 10 meters per second. So the 10 meters per second has to be on the graph. So in that case, I'm going to put every 10 boxes with a value of 2, 2, 4, 6, 8, because I'll have a 10 here. Okay? Do you all understand what happened? This is exactly how it was done also in the answers. Okay. Now, the y-axis, what is the maximum value? We did not calculate it, but let's look at the answers when we subtract. What are the answers here? The maximum value is 2.7. Let's say 2.8. Okay? We have 2.0, 2.2, 2.5, 2.7. Okay, on the x-axis, no, I don't want to look at the answers now. Now on the y-axis, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And I said I want my maximum value to be, for example, 2.8. How will I distribute that?
Every 3 boxes, 1.
Every 3
Every 4
boxes,
boxes.
1.
Every 4 boxes,
1,
1.
2, 3. Yes, every 4 boxes, 1, which is exactly what I was doing. 2.5, 2.0, 2.5, 3. Okay, now this is here we ensured that the maximum value is more than half the axes. Now this means that every small box has what value? You will divide 0.5 by 10. Okay, so you know the value of every small box. Okay, we don't have time here to, I will not give you time here to plot your graph, but I want to just stress on something. When you plot your graph, it either has to be in X's or a circle in a circle. Let me just show you 1 or 2. Let's plot 2 together. When the wind speed was 0, let's look here. When the wind speed was 0, okay, the loss in mass was 1.9. So in case you don't know how to plot your graphs and you're too embarrassed to ask, when the wind speed was 0, the loss in mass was 0.9. So I'll go to 0.9. 0.9 is how much we said?
1.9 يا miss.
1.9. Okay, I did not memorize the answers. Thank you for telling me. 1.9. Where is 1.9? Every small box has a value of how much?
0.05.
Okay, so this is 1.5, 0.05, 1, 2, 3, 4. This is 1.6. 1. Wait, 0.05, 0. You said 0.25
تحت التوبيك
or 0?
مين؟
Excuse me?
تحت التوبيك مين؟
Yes. Okay, good. This is 1.9. So when I plot this point, either I make a circle inside the circle or an X. Don't just put a point. They don't like that. And then you join the dots with the ruler. And this is the final graph. Okay? Look at this graph is not given in the mark scheme. This was done and we have it here for you. I want you to look at how they will grade you. Let's read this together. The axes are labeled with units, even scale, and plots to fill more than half the grid. Plot 5 points at least correctly. Plot 4 points correctly, then you get 1 mark. 5 points, you get all the marks. Note, plotted points must be accurate to half a small square and then straight line. You have to join the dots with a ruler. And the graph will look like this. Now, what is this? You will ask me, what is this? Because they will ask you in the next question, use your graph. Use your graph. Then you have to use your graph to determine the loss in mass at a wind speed of 10 meters per second and show on the graph how you obtained your answer. So you will go to 10 meters per second and you will move vertically with your ruler until your graph stops you. And then you will move horizontally so that you get the loss in mass on the y-axis. Now, why is this a dashed line? Because this was not part of the original graph. We extended this line so that we can know the loss in mass when the wind speed was 10 meters per second. Did you see this? So when I finished my graph looked like this. It ended at 8. This is how my graph ended. In the next question, they will tell me, use your graph to determine the loss in mass at a wind speed of 10 meters per second. Okay, I'll go to 10 meters per second and I'll find that there is no graph. So what do? I'm going to extend my original graph with a ruler so that when I go to a value of 10 meters per second and I move vertically upwards, I will find that there is something stopping me. When it stops me, I will move horizontally to get a value on the y-axis, and this will be the answer. Did you see this graph? This graph is a new example because you had to extend the graph a little bit in order to answer the next question, which is triple I. Did you see that? Are you all with me?
Yes.
Good. That's why I
دكتور
tell
بس هو
you.
اللاين نفسه didn't occupy more than half of the graph.
How come? The line, it's not that the line should occupy more than half of the graph. The values on the grid, I extended my values so that the maximum value is beyond half the grid. The maximum value 8 is here, and here the maximum value, which was 2.8, I think, it is up there. Okay? So I keep on telling you, paper 6 plans helps you practicing all paper 6 concepts. Nothing is not in paper 6 plans. Now, this question also is important. A student criticized the results by saying that the loss in mass does not allow for a fair comparison between the leaves. Suggest a more appropriate calculation and explain why it gives a fair comparison. 2 marks. Do you understand the question? They're telling you here that the loss in mass is not good enough. This loss in mass does not give you a fair comparison. Why is this not accurate? Why is this not fair? There's something wrong here. Why did we not approve of the mass of the leaf at the end minus the mass at the of the leaf at the start? This is not good enough. Why? What should have we done?
We should have calculated percentage change.
Very good. We should have calculated the percentage change in mass, not just the change in mass. Why?
Because leaves have different surface areas?
No, they have different starting mass. Why do we always calculate the percentage change in something? Because always the starting mass or the starting value is not the same. Look at the mass of the leaves at the start. One was 5.7, one was 5.3, one was 5.9. It is very difficult. It is actually impossible to get leaves with all the same starting mass. Maybe they're close, but they will never be 100% the same starting mass. So to compensate for this error, this is an error. To compensate for this error that is difficult to avoid, we calculate the percentage change in mass, not just the change in mass. And this is why we always calculate the percentage change in something, because the starting values are never the same. Okay? This is also a very important question. We always used to calculate the percentage change in mass. And I remember mentioning why we do that, but obviously now we will stress on the reason why. We will calculate, so answer with me, we will calculate the percentage change in mass, not just the change in mass. And write the equation, final mass minus the starting mass, okay, divided over the starting mass times 100. Now, this is a suggesting more appropriate calculation. Explain why. Why? Because the starting mass is not the same for all leaves. Compensate for this error to obtain accurate results. But don't just write accurate results. You need to elaborate. Okay? So to compensate for this error. This is also a very important question. Let's look at the mark scheme. We will calculate the percentage change in mass and we will give the formula because we have different original mass that will affect results to take into account the starting mass. Is this question clear? Also, please mark this question with a marker. Let me see if I have a marker here. No, here's my marker. If I am you, mark this question because this question is important. And also mark this graph because this graph is important. This graph here, it caused you or it forced you to make an extension so that you answer the next question. And also please mark this question, the area question, so that when you come to revise on the night of the exam before paper 6, you revise these questions. The area, okay? The area, the question that will force you to extend the graph. You will only extend the graph in certain situations. Why do you calculate the percentage change? This is important. Okay. Now, are you all with me? We are page 248. Now here, another picture. Draw a large label drawing of the cell X. So I'm going to draw it with you because some people have issues drawing. Let me draw it with you. I want to draw this cell. This is a single cell with chloroplast. They told you it contains chloroplast. Now this cell has how many sides? 1, 2, 3, 4, 5, 6. And of course, this is obviously a thick cell wall. So I'm going to do it here. 1, 2, 3, 4, 5, and 6. I don't want a broken line here. Oof, it's gone. Okay, again. 1, 2, 3, 4, 5, and 6. No broken lines, okay? And then I'm going to make a double line to show my cell wall. No broken lines, no shading. Good. Now I have here 1, 2, 3 chloroplasts, so I'm going to draw something like this in this area. 1, 2, 3, and I have 1 here. I have 3 here that are close to each other. And I have 1 here. Okay, so I will draw something like this. Good. I hope you were drawing it with me. Now let's see how will they label. This is a very good drawing, by the way. Now look here, lines clear, single, unbroken, no shading. Central cell has a minimum diameter of 56 millimeters, double. The detail one, double line. The cell wall has to be a double line. Any cell wall has to be a double line. And the second detail, minimum of 5 starch grains around the perimeter of the cell, and label 1 starch grain. They definitely asked you to label 1 starch grain. We missed that. So I'm going to make 1 ruled line without an arrow, and I'm going to say starch grain. I hope you're all drawing it with me. Are you all with me? Okay. Now, this question, I'm not going to be able to do it with you on my laptop, but what will you do? You will measure the length of line AB, and they are reminding you to measure it in millimeters. I will measure it in centimeters. And how do I convert it to millimeters?
Times 10.
Times 10.
Times 10. And then I'm going to calculate the actual diameter of the starch grain using your measurement for line AB, the information in figure 1.2 and the formula. So let's look at the information in figure 1.2. They gave you the magnification. So you will measure this. I don't know how much this will be. Let's say it is 10 millimeters, for example. And you're given the magnification. So you will go and apply that on the formula. And even if they don't give you the formula, you already know the formula, which is I am. You have the magnification. You have the size of the image from the picture, and you will be able to calculate the actual image. Give your answer to 2 significant figures. So you have to round. If it's a decimal, I want 2 significant figures. I don't know what's the answer, but I showed you how it's done. This is 3 marks, so we don't want to waste any mark for any reason. Okay. Again, I think you will be asked to draw. Draw a label, a large diagram of the holly leaf. Again, drawing for the 10th time. Don't tell me I don't know how to draw. You will draw whether you like it or you don't. This is the central vein. And then let's count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 9 on one side. I'm going to try to do this. 1, 2, 3. This is 4, 5, 6, 7, 8, 9, and then try to do the same on the opposite side. Okay, my drawing is not very pretty. And you have a vein here in the center. And 1, 2, 3, 4, 5, 6. So I'm going to do 6, 1, 2, 3, 4, 5, 6. Why am I counting? Because I want to show you that you have to keep the same details. It has to be a replica of the picture. I hope you're all drawing with me. Drawing will look something like this, okay? I got this picture from the internet. I found something that is similar and use more than half the space available, etc., etc. And the same thing, I'm not going to do this with you. Again, you will measure in millimeters and you will follow the instructions to get the magnification. The same question. Now, I want to go to this question. Maybe this will be the last one I will solve with you for today. Here, students investigated the relationship between the number of prickles on holly leaves. What are the prickles? These are the prickles. These are prickles, okay? So they counted the number of prickles on holly leaves and the height of the leaves above the ground. Now, this question is tricky. The students collected leaves from a total of 5 trees. The students took leaves from 7 different heights on each tree, and they collected 10 leaves from each height on each tree. They counted the number of prickles on each of the holly leaves and calculated the average number of prickles per leaf at each height. Okay, now why did I mark what I marked? Because I have 5 trees. How many leaves did they get from each tree? 10 leaves. So what is the total number of leaves?
50 leaves.
Excuse me?
50 leaves.
Yes, good. 50 leaves, because I will need it now. The results of the investigation are shown. So this is the height and the average number of prickles per leaf. The students counted a total of 614 prickles on the leaves collected at a height of 2 meters. So we will go to 2 meters, and of course, they will ask you to calculate the average. How do we calculate the average? Calculate the total number of leaves collected at 2 meters above the ground. How many leaves? Total, we just did it. We have 5 trees, and we took 10 leaves from every tree. 5 trees and 10 leaves from each tree. So how many leaves in total? You just said it.
50?
50, yes. This question, when I got it in the mock exam last year, a lot of people did so many mistakes. So just read the question. You don't read the question carefully. You have a total of 5 trees, and I took 10 leaves from every tree. So the total number of leaves is 50. Now calculate the average of the prickles. 614 prickles on the leaves, on the 50. So how many leaves in one, how many prickles on one leaf? If I have 614 over 614 prickles in 50 leaves, how many prickles in one leaf? And how do you calculate the average actually? Total number of prickles over the sum. Yes? Are you with me? I know that this question is not very clear and it caused a lot of problems last year. But do you now understand it?
Yes.
Okay. When we divided 614 divided by 50, give your answer to the nearest whole number. 614, which is the number of prickles on the 50 leaves. We want the number of prickles on one leaf. So we will divide 614 by 50. 1 times 614, if you want to cross multiply, 614 divided by 50. So this will be 12.28. When I want to give it to the nearest whole number, what will be the answer?
12.
12. So I'll put 12 here. Okay? Oh, yes, we're right. Thank God. Okay, good. Now, so also this question is important. Please mark it because it's confusing. Mark it. No, this is not how I mark it. Mark it. Like this, make a mark. So when you come to revise any question that you find difficult in any paper, paper 4, paper 6, paper 2, mark it with a marker. So when you come to revise, you go straight to this question. Okay, now they will tell you, state 2 ways the students ensured that they collected a representative sample of the leaves. Now, this is also a very important question. The word representative, I told you to add it to your vocabulary for paper 6. How did they make sure that they collected a representative sample of the leaves? Representative means
They
a sample that reflects,
They
a small
collected
number?
leaves from different trees.
Okay, from different trees, this can be accepted. What else?
They
Not from
collected
one
many
tree.
leaves.
Yes, this is the most important. They collected many leaves. They collected a total of 50. Representative means it represents the real figure, the real number of leaves or the real results. Okay? And you can say that they repeated more than once. They calculated average. They got the leaves from many trees. They calculated, but the most important answer that they got many leaves. Add, please, this word to your vocabulary. Again, I'm going to mark it with you. Okay, representative means I'm going to collect a lot of samples so that it reflects the real results. Now the graph, 4 marks. Let's do this and then I'll let you go. What will be plotted on the x-axis? What is the independent variable? What is on, tell me.
The height.
Yes, how did I know? It's on the left side of the graph. If I read the question, I'll figure it out. Or I can go directly to my table. It's on the left side of the table, so that will be the independent variable. And what will be my results? What will be plotted on the y-axis?
The average.
The average number. Again, I'll do the same thing for every single graph, whether line, bar, everything. I have to divide it into boxes of 5, and don't think that this is a waste of time. Without this step, you will not be able to do anything. 1, 2, 3, 4, 5 mark. I'll do it with you. Okay. Now let me see what will I write on the x-axis and what are the units. Height in meters. I'll write the height of the tree here, height in meters. And then let me look. What is the maximum value? 5. How many boxes do I have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, 1, 2, 3, 4, 5. Okay, I can do that. How will you distribute it? Or I can do 1, 2, 3, 4. No, I would do every 2
10
boxes. Yeah, every 10 boxes. I mean, every 2 boxes. 1, 2, 3, 4, 5. The 5, yes, is not at the very end, but I occupied more than half the grid. And I will do the same for, and use the decimals because it's written as decimals. So I'll write it as decimal, 1.0, 2.0, 3.0. Okay, we don't want to upset them. Now we have the y-axis. What's the maximum value? 18. Average number. So I'll write here average number, no units, and I'll do exactly the same. Okay, we said the maximum value 18. Yes, let's say 20. How many boxes? 1, 2,
Two.
3, 4, 5, 6, 7, 8, 9, 10. Yes, very good. Every box, 2. 2, 4, 6, 8, 10, 12. Now we became experts. Very good. 14, 16, 18. Perfect. It's a very perfect graph. Always know that the graph fits your numbers, your values, okay? Now I'm going to plot 2 with you so that 0.5, 18. How do I do that? Some students, they really don't know how to plot. So 0.5, this is 0.5. Let me change color. This is 0.5 here. We said 0.5, 18. So 0.5, 18. Very easy to plot. Here, I'll make a dot in a circle or an X. It's your choice. And we will continue like this until we finish our graphs. And I highly advise you, before you start plotting, know the value of every small box so that your plotting is accurate. And after you're done, join the dots with a ruler. Your graph will look like this. Okay? Obviously, as the height above the ground increases, what is happening to the average number of prickles? It is what?
Decreasing.
Decreasing. Okay? Okay. Now I want to answer question D. Let's answer this question till the end so that we finish this exam. Figure 2.2 shows another leaf count and record the number of prickles on the leaf. Fine? Use the information in table 2.1 or your graph in 2BII to estimate the height on the tree from which this holly leaf was collected. Now, this question is important, also mark it. What is the number of prickles? I'm going to count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Okay, so they said either use your table or use your graph to estimate the height on the tree from which this leaf was calculated. So I have 17 prickles. If I go to my table, 17 is closest to the number 18. So what will be the estimated height above the ground?
0.5.
0.5.
0.5.
I can use my table or I can use my graph. If I will use my graph, then I'm going to go to my graph. Where is my graph? Here. And I'm going to use my graph to estimate. This is just an estimation. Of course, the table will be easier, but I'll show you all situations. It's going to draw 1 minute. Yes. It's not drawing. Okay, now it is. Then I'll go, they said which height? Oh, yeah, they said the number of prickles, we counted them, we found them 17. I'll go to number 17 prickles, move horizontally until the graph stops me, and then I'll move down and I'll get an estimate. So I either use my graph or use the table. Okay, here, this is another question where they ask you to use the table. And then the last question, and I promise I'll let you go. Leaves contain starch. State the solution used to test for starch and give the result for a positive test. What is the solution that I'm going to use to test for starch?
Iodine.
Iodine. Okay, now I want to point out something. They only want the name of the solution, but the question can be different. They can ask you, what are the steps to test for starch? Can I put iodine directly on my leaf?
No, I have to boil the
No.
leaf first, then
It's
boil it in
starch.
ethanol.
Yes. When they
You-
ask you for the
in the water
steps,
bath.
then you have to boil in water and then boil in ethanol and then what?
Dip it in water.
Very good. Dip it in water again to make it soft. And after all this, I will add my iodine. Now, what is my positive result?
Blue black.
Blue black.
Excellent.
Orange.
It will change from yellow, brown. Please write the starting color. Yellow, brown to blue, black. Very good. Is this question finished or it doesn't finish? Okay. I'll stop here. Fine. Please finish paper 6 plants. I extended the date again so that I give you time. And if you have any question that is confusing you, take a picture of the question and send it to me on WhatsApp and I will help you. I will never ignore your question. Okay? Paper 6 plants is the best practice for paper 6. We spent 2 sessions or 2 blocks working on paper 6 in school and we worked on paper 6 online. Not because I want to waste my time and yours, because it's very important. Because starting tomorrow, I'm going to start something else. Okay? Anyone has any questions? Please make sure that you are very good at your graphs and your drawings and your tables. And if you are not sure of anything, please ask me. Anyone has any questions before we go? Okay, thank you. And I'll see you tomorrow. Tomorrow we'll start a new lesson. We'll start immunity. And I want you to get paper 4 classified because we will work on paper 4 transport in animals. See you tomorrow.